So our next talk is very special because we set up a lot of goals this morning, and we said how important it will be to increase crop productivity and quality. But I think one of the most important goals is to educate the new generation who would actually tackle these goals that we set up and try to solve them. And this is Yael Ben David, who is a master's student at the lab of Professor Avia Zilberstein in our department. And Yael's project is on improving proteins in plant by trying to manipulate the contents of amino acids. And as we will hear from Yael, this is a very difficult task, but Yael has done some very nice progress, and we are happy to hear her.
Thank you very much. It is my pleasure to be the closing talk of this day in the Food Security Conference. The title of my work is basically pretty complex, but what we're going to do during this 15-20 minute talk is to be the detective and find out how we solve things in the scientific lab at Tel Aviv University. Okay. All right, so I think everyone who's been here today is basically convinced that food is important and there's not enough of it to go around. So I don't really need to convince you of that. Just to say that one nutritional value that is very important in our diet is amino acids. They're what build proteins, and there are a few of them that are essential in our diet because we can't produce them ourselves. We have to have them from plants. So plants are the primary producers, and that's where we get our amino acids from. I keep pressing this. So the first question is, okay, we get the amino acids from plants. How do plants produce these amino acids? So this question is basically resolved. We know this already. The plant cells have different enzymes that convert amino acids such as aspartate into other amino acids such as lysine and isoleucine and etc. So we know these different pathways exist in the plant cell, but the next question that we need to ask is, how is this production regulated? And this is not known. So in general, the genes in the plant are not all going to be expressed at the same time. The plant needs to know how to differentiate different conditions. For instance, in darkness with low energy levels, different genes need to be induced, rather than in light, when they have plenty of energy, other genes need to be induced. And the regulation of these conditions are done not at the level of DNA, which exists all the time, but at the level of RNA, which is what will later be translated into proteins that will do the job of converting aspartate, for instance, into all these different amino acids. So the regulation we're talking about is at the level of going from DNA to RNA, which is the transcriptional level. So what regulates this process are transcription factors. And simply speaking, what they do is basically an on-off switch. How do I go back now? It's basically an on-off switch at the level of the DNA sequence. So biology, as in biology, nothing is absolutely 100%. It's not a toggle switch, but it allows us to either upregulate the levels of the gene we're talking about, which is aspartate kinase, this first gene here. So we can either upregulate it or downregulate it depending on the different conditions. Okay, so this is where it starts to get a little bit complicated. We said that the different transcriptional regulators work through the promoter area of the gene. So our question is, who does this? Who binds to the promoter of aspartate kinase in order to regulate its production? So what we used is a system called EastOne Hybrid. So I'll just ask you to bear with me during these few minutes and try to understand how the system works. The basic idea is that we have a transcription factor that is created out of two different domains. One is an activation domain, and one is a binding domain. In the binding domain, we can put basically any protein that we want from the plant. For instance, a protein that is involved in, say, photosynthesis. So that protein would not bind to our promoter, which we would use the aspartate kinase promoter. So if it does not bind to the promoter, we would not have induction of the reporter gene, and we would all see these yeast colonies as white colonies. But in the case of a protein that does bind to our promoter, because we're looking for a protein that would bind to the aspartate kinase promoter, if it does bind to it, then we would have activation by the activation domain, and we would see a blue colony. So a blue colony basically means we found a protein that binds to our promoter. And this is exactly what we did. We took the aspartate kinase enhancer, which is actually part of the promoter, and we found that the protein in the binding domain that gave us a blue colony was called DPBF4. So we found that DPBF4 is a protein that can interact with the promoter of aspartate kinase. So we're thinking perhaps DPBF4 can regulate the expression of aspartate kinase. So who is this DPBF4? It belongs to a family of transcription factors called B-zips. And the reason they're called this is because they have a leucine zipper, which is an actual kind of sort of zipper structure. And the second part of its structure is DNA binding. So this structure here is a predicted structure. And the reason it's only predicted is because we get that structure from humans, actually. Because B-zip transcription factors are transcription factors in humans as well. And there's a famous pair of transcription factors called C-Fas and C-Jun in cancer research. So thanks to the cancer research, we have a predicted structure of what these B-zips look like. So DPBF4, we know, binds to this promoter sequence. So back to B-zips in plants. There are many B-zips in plants. In fact, there are 75 of them. And they are grouped into different letters, basically. The DPBF4 is from group A, and it belongs to the ABI5 subfamily. The reason it's called the ABI5 subfamily is because ABI5 is a very big transcriptional regulator in plants, and I'll just ask you to remember the name ABI5 for later. It is part of the this subfamily here, and all of these basically have a similar structure and are homologous to one another. Okay, so back to aspartate kinase. We found a protein that might be the one who regulates it, and we want to see how aspartate kinase behaves. So in darkness, we see that it goes down. The level of RNA, or transcription of aspartate kinase, is downregulated, as opposed to light, where it is upregulated. And this makes sense. No light means no photosynthesis, means no energy, so there's no reason to give energy to make amino acids. So how does DPBF4 behave in the same conditions? So DPBF4 basically does exactly the opposite. DPBF4 is downregulated in light and upregulated in dark. So this means that if DPBF4 goes up when aspartate kinase goes down, perhaps DPBF4 is a repressor of aspartate kinase. So next, how do we find out what DPBF4 does? Well, biologists like to do just different kinds of experiments. For instance, overexpression or having DPBF4 in abnormally high levels. And when we did this in our lab, then we found out that really nothing happens. We don't see any difference between the wild type or the regular plant and the version that has the abnormally high levels of DPBF4. So the next step was to knock out DPBF4, as in not have hardly any levels of it in the plant. And this also did not have much of an influence. We can see that there's also no difference between the wild type and the version that has hardly any DPBF4. So you're probably thinking now, okay, you found DPBF4, you know that it binds to the AK promoter, but it really doesn't do anything. So now what? So we know that DPBF4 does not function uniquely, obviously, because when we either put a lot of it or put none of it, nothing really happens. So perhaps there's a redundancy through other DPBF transcription factors. The family before that I showed you, ABI5, has many different homologies to DPBF4. So perhaps they play a part in aspartate kinase transcription regulation. So we know that there is redundancy of B-zip transcription factors. How do we overcome this? So before I get to answering that question, I'd just like to go over a little bit the structure of B-zips and how they work. So as I mentioned, they're called B-zips because they have a leucine zipper structure here. And they have a basic region that binds to the DNA at their N-terminal. So they can work either as homodimers, meaning they have two parts that are identical. For instance, the green would be DPBF4, and the blue would also be DPBF4. But they can also function as heterodimers, meaning you could have DPBF4 as the blue, and perhaps ABI5 as the green. So this gives us a lot more combinations that we would have to deal with and understand how they work. So what we did to overcome this redundancy is use a dominant negative system, meaning something that would act as an evil magnet to take all of the B-zips that act on the same way and basically incapacitate them. And we used something called SRDX, which is a sequence of amino acids that would give the B-zip that we would put it on a dominant negative attribute. And if you're not understanding exactly what SRDX is, that's okay, because we have a few slides coming up to explain it better. So DPBF4 has a sequence called SRDX on the end of it. And as I said, it acts basically as a magnet. So anything that would bind together with DPBF4, for instance, ABI5 binding to DPBF4 SRDX, would be magnetized to it and not be able to work. So we want to see if this is actually the case. So we know that DPBF4 binds to the aspartate kinase promoter. And then when we put SRDX on it, then we see that it does not bind anymore. We have the blue colonies for DPBF4 and just white colonies for the DPBF4 SRDX. And what we also see is in EastOne Hybrid, which is a system not of trying to see how a protein binds to DNA, but rather how two proteins interact with one another, we see that the strongest interaction that we have is when both of them are DPBF4 SRDX, meaning that perhaps this interaction here at the leucine zipper makes a very, very strong interaction, and perhaps this strong interaction is what opens up the binding domain, doesn't allow it to interact with the DNA as we see from the EastOne Hybrid of the white colonies. So the next question is, okay, we see that it works in yeast. What happens in plants? So we use a system called split YFP, and once again I have to briefly explain how split YFP works. There are basically two halves of a fluorescent molecule, and when they come close enough together, then they fluoresce. So what we did is put one half on one of the units of the dimer, and another one on the other half, and indeed we see that there is an interaction, all these yellow dots there. Another experiment that we did was taking heterodimers, meaning, for instance, DPBF4 and ABI5, but what we did this time is take one half of the fluorescent molecule on the C-terminal and one half of the fluorescent molecule on the N-terminal. So we would actually expect not to see any kind of fluorescence here because they would be too far apart. So what this tells us is that the predicted structure we have from the cancer research is not exactly right for B-zip transcription factors in plants. Perhaps this folds over and somehow the N-terminal gets more closer to the C-terminal, and that's how we see the interaction between them. So what happened when we used DPBF4 SRDX as one of these units was that you can really see a depletion in the fluorescent molecules. And this further strengthens our theory that SRDX does make this structure stronger, but doesn't interact at the N-terminal. So the way to sort of animate this is like a inverted tweezer. So this inverted tweezer would work this way, and normally it would bind to DNA. And when we have the SRDX, because of the tighter bond, then it would no longer bind to DNA. So we can use SRDX as a tool for identifying B-zip targets. For instance, any BZIP tested that we know interacts with DPBF4 would regulate this DNA and whatever gene it is accountable for. On the other hand, if we put DPBF4 as RDX and incapacitate this, then that gene would no longer be regulated. So if we find genes that have changed expression, then we know that they must be regulated by these BZIP transcription factors. So we know this is the case for aspartate kinase. When we did use DPBF4 as RDX, its levels went way up. Before, we said that DPBF4 is a repressor of aspartate kinase. So once we take DPBF4 out of the picture, aspartate kinase can go back up again, and that's exactly what we see. I forgot to mention that's our modeled plant called Arabidopsis. All the plant scientists probably know that, but all other people, you should familiarize yourself with the name because it gets used a lot in these fields. So that's our Arabidopsis. So the question is, how widespread is the influence of BZIP transcription factors? We know that it influences aspartate kinase. What else does it do? So don't be alarmed. The answer is it's quite widespread. All these red arrows, basically, are places where BZIP transcription factors affect the regulation of all these different genes. So back to the aspartate pathway, and that's what we were looking at on one of the first slides. We know here that BZIP transcription factors affect this pathway as well. So back to sum this whole picture up. What we asked in the beginning was, how is the production regulated? And we know that BZIP transcription factors act on the level of transcription between DNA and RNA. And the question is, does it actually do anything at the protein level? Would we see any difference in amino acid actual levels? And the answer is yes. The orange dotted lines are where we use DPBF4 as RDX, as opposed to no DPBF4 as RDX. So we do see that there's an influence of using it at the actual level of amino acids. That's another just to let you know that DPBF4 as RDX indeed has a major influence on actual amino acid levels in plants. So that is the answer to our first question of how does the plant regulate amino acids, and at least a partial answer is through BZIP transcription factors, which are also, actually we have them in cancer research. So that's it. Thank you very much for listening. I hope you enjoyed the first day of the conference. Thank you to all the members in the lab, and that's it.
Thank you very much. So the world belongs to the young, and the big scientists, and Yael, if anybody has questions.
Have you been able to make a mutant plant that would have increased amino acids?
These data came like a week or two ago. So this is really, yeah. Yeah.
Any more questions to Yael? Okay. Thank you very much. And thank you.